Now, this next little module, we're going to focus a little bit on research designs and approaches to genetic epidemiology. There are several useful terms that would be useful to keep in mind as we go forward from here on. There are three that I will use quite frequently. The first is genotype, and that is, in fact, nothing more than the genetic makeup of the individual that we're about to study. The second is phenotype, and that is if you would, the physical characteristics, the clinical appearance of the particular trait of interest to us. That could be alcohol dependence, it could be major depressive disorder, etc. And the third term is the proband. And the proband, in fact, is the index case when we're studying a large pedigree or family history. The person who is the proband, in fact, could be affected with alcohol dependence or other substance use disorder, or in fact, they could be unaffected and may have a particular trait that is not very obvious to us, such as sickle cell anemia or another such type of characteristic. In terms of psychiatric genetic epidemiology, there are really two basic approaches that we focus on. One is the phenotypic examination of individuals in our group. And as I said previously, these are really focused on the observable manifestations of a particular trait. And we assume that these manifestations reflect some underlying genetic vulnerability or the action of several gene or genes. The phenotype, in fact, does provide a very crude estimate of genetic effects. The second approach is a genotypic approach, in which we characterize the individual or the trait in relation to its genetic bases. And in current methodology, we have a variety of ways of doing that. Two of the most common, in fact, include either doing what's called a GWAS, G-W-A-S, genome-wide association study, in which we look at the entire human genome in relation to the trait to identify associations of specific genes with that particular trait. The second approach, now becoming more frequently used, is that of deep sequencing, in which we do identify a gene and then, in fact, try and identify more, if you would, the makeup of that gene in terms of single nucleotide polymorphisms. Those are beyond this particular lecture, but you should be familiar with the terminology. When we think about a particular trait and whether it's genetically determined or not, there are some very basic approaches that are used across psychiatry and as well as other medical disorders. Evidence for a genetic trait typically comes from three sources of information. The first is the study of large families, typically multi-generational families, and we will look at that visually, trying to determine whether a particular trait, such as alcohol dependence, is in fact transmitted across generations, from parent to child to grandchild, etc., even into additional aspects of the family tree, such as nieces, nephews, etc. The second approach where we get the information on genetic basis is the study of adoptees. And as I indicated earlier, with adoptees, we have good information about where their parents were affected with a particular trait or not, such as alcoholism, alcohol dependence. And for those individuals who are then adopted away into homes in which that is absent, we would anticipate that the trait would in fact be either absent or very low vulnerability or very low expression. The third source of information is the study of twins. And as you know, there are monozygotic twins, in which are identical genetically across their entire genome, and fraternal twins, or DZ twins, which have share 50% of their genes. And it is this combination of studies that we use to determine or consider whether a trait in fact is genetically determined or not. There is no single method currently available to clearly determine whether in fact a particular trait is genetically determined. So with family pedigree studies, as I indicated, we typically start with a very large pedigree. We try and make sure that it is in fact multi-generational, that there are 2 and hopefully 3 generations included, and as many of the biological relatives are assessed as possible. And when we have a positive effect, we can then say it is a genetically determined, and this has to be in the absence of particular nationalities. There may be some issue with other psychiatric disorders, very common in substance abuse situations. It should or may vary a bit by gender, but should still not be absent from one gender to the other. And the severity of the disorder may in fact, in the parent, may moderate the heritability. A moderately or lightly affected parent, for example, is likely to convey very little genetic vulnerability to their offspring. However, a severely affected parent typically conveys a significant amount of genetic vulnerability to the offspring. Consequently, the more the parent has, the higher the likelihood of the child being affected. But with family pedigree studies, of course, as we've been saying, you really cannot separate the genetic and environmental sources of influence. So we need to move on to other types of information to further document whether there's a genetic factor involved. And with that, one of the best ways of doing that is through twin studies. Typically, what we will find is that in monozygotic twins, the concordance or the rate of similarity is very high. Thus, if one of the twins is affected with alcohol dependence, we would expect, if in a genetically determined trait, that the co-twin would also have a high degree of likelihood of being affected. In dizygotic twins, where they share only 50% of their genetic makeup, the concordance rate should be quite a bit less. So that when we compare the monozygotic MZ versus the dizygotic DZ twins, if it's a genetically determined trait, we would expect the MZ twins to have a much higher rate of alcohol dependence. And this, of course, does assume that the twins are raised in a similar environment, which we know does vary from family to family, and that in fact that their course of development is very, very similar. If there's variation in that course, if one is separated and raised in another family or another type of environment, then that would influence the concordance rate. Adoption studies were very natural experiments and were in fact very popular in the 1940s, the 1950s, and the 1960s, when adoption was much more prevalent than it is today. But in these natural experiments, again, one of the things we would expect is that individuals born of an affected alcohol-dependent parent, regardless of the environment, would also show an increased rate of alcohol dependence compared to the general population. Most of these studies were in fact conducted in Scandinavia, where birth records and public records are well known and well documented. The only study in the United States of adoptees was conducted in Iowa and used birth cohort information from Catholic Charities, who were very active in adoption services in the 1950s and the 1960s. But let's take a look at some data to kind of demonstrate what the points I've been trying to make. And in this particular slide, you can see that the rate of alcoholism or alcohol dependence among relatives of alcohol-dependent probands or index cases. In this case, these probands, in fact, were treated on an inpatient service. But compared to the general population, what is the rate of paternal alcoholism in these alcohol-dependent patients? You can see that paternal or having an affected father, the rate was about 37% for males, and among females, it was only about maybe 15%. When you look at maternal alcoholism, you can see the effect again is a bit gender specific, with more alcohol dependent female probands having an alcoholic mother compared to an alcoholic father. Getting to the issue of twin data, we see in fraternal twins that in fact the rate of alcohol dependence is pretty similar in fraternal twins, but when you get to identical twins, it is much greater among males than females. Again, you see a gender effect here, but the genetic indication is still very, very prominent. As we look at the literature, look at our own studies, there are a number of real challenges that we should keep in mind when we're trying to determine genetic vulnerability. The first, in which we've mentioned previously, is that of clinical heterogeneity. In fact, all persons with alcohol dependence do not look alike. There's significant variability in terms of ages of onset, in terms of the symptom picture, in terms of the clinical course, and even in terms of treatment response. We also know that many people with alcohol dependence have significant psychiatric comorbidity. This could be comorbidity on another substance. Tobacco and marijuana are very frequently seen in individuals affected with alcohol dependence, as is psychiatric conditions such as comorbid depression or antisocial personality or behavioral disorders. There's also variations in diagnostic criteria used when you look at the literature. In Europe, you'll often see papers that cite ICD-9. In the United States, the differences in ICDs could be 9 or 10. In the United States, you might see differences in relation to the use of the diagnostic statistical methods, the DSMs. And this could be DSM-2, 3, 4, 5, as is currently. Or there have been times when different agencies, such as the National Council on Alcoholism, have developed their own criteria. So we have to be careful to be sure when we're reviewing the literature that in fact the criteria used to identify a particular case or a proband is very much the same. Similarly, when we look at different sources of data, it could come from direct interview, it could come from families or other individuals reporting on individuals, or it could come also from data that is obtained clinically or through records, state records. So again, there's variations in the quality of the data that we may obtain. Certainly in terms of sample size, some samples that we see in the literature are very small. More recently, in epidemiological studies, we're now seeing sample size in the tens of thousands, and before very long, we're going to see sample sizes in the hundreds of thousands. And again, the composition of those samples, what's the rate of affectation, what is the gender composition of that sample, and also what is the racial ethnicity of that sample. For example, in different parts of Asia, there are genes, in fact, that are protective of persons such that they are not able to drink alcohol without consequence. And consequently, the information we have about the population prevalence rates, responses to treatment, etc., will vary quite a bit. Again, depending on the ethnicity of the individuals. Similarly, ages of the proband and their biological family members at the time of assessment is quite important, as there is an age of risk, if you would. Most people with alcohol dependence will develop that condition sometime in mid to late adolescence, and certainly typically not later than the late 20s or maybe about age of 30. There's also a variety of environmental factors, and that has to do with the availability of alcohol, the taxation laws in that particular country, and potentially also in relation to the policing efforts with respect to people who display alcohol consequences in public. How can we tell between genetic and environmental factors? Again, there are some things to keep in mind as we go forward. First is that genetics really does not equal predestination. Unlike with hair color or eye color or maybe even stature, individuals who carry a genetic vulnerability for a particular psychiatric condition, including alcohol dependence, are not necessarily predestined to develop that condition. And that's something that is very important for us to remember. Offspring, not all offspring of an affected parent will develop that condition. We also should keep in mind that there are genetic factors that are related to an increased vulnerability for developing the disorder. That is, factors in the genome that increase a person's risk, and we'll talk about those in the next module, as well as genetic factors that may, in fact, limit the vulnerability for developing alcohol dependence. And these are factors that, in fact, that may have to do with the metabolism of the drug, the taste sensitivity to the drug, etc. Keep in mind as well that alcohol dependence is a complex trait. There are a variety of different genes, and I'm not sure exactly yet, nor is anyone else in the field, exactly how many genes that are involved with this. A number of studies are now suggesting that it could be somewhere between 30 and 50 genes, individual genes, contributing a small amount to the overall vulnerability. Also keep in mind that the alcohol dependence from a clinical presentation is very, very heterogeneous. Thus, we can say then that maybe even though there may be a panel of 30 to 50 genes affecting the vulnerability, that only some of these come into play at any one time. And then these, in fact, may relate to the clinical presentation. Finally, there certainly are a variety of psychological and environmental factors, which we'll talk about in the next section, that in fact do relate to the activation or the sensitivity of specific genes for an individual. What I'd like to show, though, is really a diagram that was first presented by Dr. Cloninger in the 1970s. And what it does is really show the interplay, the interaction between genetic load and environmental factors. And over on the left-hand side, you can see here that individuals with low genetic loading and low environmental factors or sensitivity have really a very small likelihood of developing a particular trait like alcohol dependence. And again, it's more of a almost a reverse Poisson type of distribution. That is, genetic load increases and or the environmental sensitivity increases, the probability of developing alcohol dependence does increase. and does reach a bit of an asymptote, if you would, as the genetic load and the environmental sensitivity, in fact, come together, such that the likelihood of developing alcohol-related problems is close to 100%. We also should keep in mind, and it's a bit of a puzzle yet as to why this is, but in fact the probability of developing alcohol dependence does vary by birth cohort. And this is very likely is due to a variety of environmental factors. In this particular graph developed by Bridget Grant, one of the things that we can see is that individuals born in the late 1800s, early 1900s had a cumulative probability of developing alcohol dependence that was much lower than individuals born in later birth cohorts. Why is that? Well, think about this from a historical perspective. In fact, there have been 2 world wars that came into play, to say nothing of the Korean conflict and the Vietnam conflict, as well as Prohibition. Typically following a major armed conflict or war, there is some level of reduction in terms of availability of different types of goods, and alcohol was one of those as well. Also, as you look at the 1968 to 1974 birth cohort, these individuals came into society at a time when drinking in public was much more common, much more accepted. There was much more in terms of gender equality, and women were typically much more likely to be in the workforce during that particular time. Even with all of that, if you look at where these lines hit an asymptote, it is almost always, regardless of birth cohort, in the late teens, early 20s, when the curves will asymptote. There's only a slight increase thereafter, and that is true across all of the different birth cohorts. Again, showing that the gene pool has not changed at all in probably more than 1 million years, but there are environmental influences, in fact, that do impact the prevalence and the course of alcohol problems in the humans.